that Facebook group and you want the link, then email me and I'll be glad to send it to you. Right now, there are probably about 10 different lectures that I posted up there. Keep in mind, they're not all from your class. Some are from the other section, so you're welcome to listen to any of them. You'll get insights from any of them, and sometimes they, like you all, get me running down rabbit trails, and then you'll hear some of those rabbit trails along the way as well. So, the cell makes enzymes. Any cell makes enzymes. And the reason they do is because they want to be able to catalyze chemical reactions very quickly with less energy input to catalyze those reactions. Presumably that's the driving force for having enzymes. And so now we need to focus a little bit and look at enzymes. And you make all these proteins, and most enzymes are proteins, and you make them, and they have some function, and they're going to do that reaction as quickly as possible unless there's some way to control that enzymatic function. And one way to control that enzymatic function is this process called allosteric regulation. So we got to break down this word allosteric real quickly, and let's think about this half first, the steric part. What does steric mean? It means conformation, it means shape, it means how it's structured. And so we're talking about something about enzyme structure here, and then we've got this allo part on the front. What does allo mean? Do you have an idea? Other. So together it means other shape. You already know that enzymes have a particular shape that they have to fold into to be able to function. And an allosteric regulation process causes that enzyme to fold into another shape, a non-functional shape. And here's how it works. So this enzyme that's depicted on this page is an allosteric enzyme. It's got the active site where the substrate can interact, and that's typical for every enzyme, right? But this type of enzyme, these allosteric enzymes that are they're regulated by allosteric regulation, have a second site. And here, this site on this side right here is called the allosteric site. It's not the active site. It's not where the reaction is going to be catalyzed. It's a different part of the enzyme from the active site. And what can happen then is in the normal reaction, the substrate goes and interacts with the active site, and the enzyme will catalyze that reaction. But with an allosteric enzyme, some allosteric inhibitor will come, and the allosteric inhibitor will interact with some other part of the enzyme and cause that enzyme to fold into a conformation that's no longer functional to catalyze that original reaction. Does that make sense? So you've got something else, and we call that guy an allosteric inhibitor. Some other chemical, some other molecule that can interact with a different portion of the enzyme and be able to interfere with the activity of that enzyme. So in this beautiful pathway that's right here at the bottom of this page, we've got a three-step process in this pathway. And if this was a pathway where some substrate was feeding into it, the cell's got to make that substrate or bring it into the cell. And so it's got that substrate, and once it's got that substrate, it can feed it down this pathway, and some end product is going to be produced. But pretty soon, if that end product is no longer needed, in other words, if it's not being used for whatever it was originally designed to do, the concentration of that end product is going to go up and up and up, and it's going to increase in the cell. The cell's not using that end product as fast as it was before, and it doesn't need that end product anymore. So the cell needs some way to tell itself, hey, I don't need that product anymore. It's no longer useful to me. We have way more than we need. We have plenty of that product. And one way that the cell can do that is if this end product then can feed back and interact with enzyme A, the first enzyme in the pathway. And most of these allosterically regulated pathways are controlled at the first enzyme. There's a few that are controlled at enzyme B, but most of them are controlled at enzyme A or somewhere early on in the pathway. Because think about it, this substrate, it took energy to make this substrate, and if it's getting pumped down here and there's no use for this end product anymore, the cell's not using it and it's accumulating, then energy is being wasted. That energy could potentially be funneled down a different pathway, and the cell then would be able to not waste the energy in this original pathway, but use it in a different pathway where it can be used for something else. And so that's called allosteric regulation. Usually the end product is that allosteric inhibitor that interacts with the enzyme that's early in the cycle. So think about this. We're making this product in this pathway, and this end product is increasing in concentration, and we're getting lots of it, and so this pathway gets shut off at enzyme A. Everybody good with that? Enzyme A is now blocked, and the way it's blocked is because that end product is binding to that allosteric site and inhibiting that enzyme, so it can't run anymore. So pretty soon the cell's still doing metabolism, and the end product concentration begins to go down. As it goes down, there's no more of this allosteric inhibitor to bind to the enzyme, and so as soon as that allosteric inhibitor releases, we go backward to this conformation. Now that enzyme can become active again, and as soon as that happens, the pathway can begin to run again and produce more of that product. And so the cell can nicely control the amount of that product and the amount of the substrate that's feeding down that pathway as it's looking at the amount of the final product that's in the cell. So that's allosteric regulation, and there are a number of pathways that are controlled that way. I told you earlier that glycolysis is constitutive, and almost all cells are going to have those enzymes just functioning straight down. They're not going to be controlled by this process. But some of the other processes that we've talked about actually are controlled in this way because they can move that energy off to a different pathway if they're no longer needing that particular product that the pathway produces. So that's allosteric regulation, and it's important to the cell because everything the cell does costs energy, and the cell needs to be able to direct that energy in a certain pathway that's going to be beneficial to the cell. There are other ways to inhibit enzymes, and this is one that you're already familiar with. Some of you have gotten an infection, maybe a sinus infection or a throat infection or an ear infection, or possibly a few others, and the doctor gave you a sulfa drug. You don't have to raise your hand if you did that, but you were taking advantage of this type of inhibition. If you've ever taken a drug and the doctor said this is a sulfa drug, and you actually took advantage of this. This is another method of enzyme inhibition. And in this case, we're talking about competitive inhibition. And in this case, we're not looking at some other site on the enzyme. We're actually looking down at the active site. And in this first panel here, they show you that this substrate interacts with this enzyme. And the substrate that we're talking about is called PABA. That's a nice short abbreviation. The full name is para-aminobenzoic acid. And some of you guys are chemists, so you can look at this part right here, and you'll see para-aminobenzoic acid. This ring right here is a benzene ring, and it's got an amino group attached to it at one end, so that's the para-amino part. So this is para-aminobenzoic acid. This part up here is a carboxyl group, and that's an acidic group, and therefore the name. Para-amino, that's talking about this, benzoic acid. You couldn't see anything that I just did there. Para-aminobenzoic acid. Okay, that's how it gets its name. All right, so para-aminobenzoic acid is made by bacteria. This pathway that we're looking at is actually a pathway that's involved in nucleotide biosynthesis, building those building blocks that are needed to build DNA. And so, remember, we talked about E. coli. You can feed them E. coli, you can feed them nitrogen, you can feed them phosphorus, you can feed them all those major and minor elements. They can make everything that they need. So they've got to be able to make the building blocks to build DNA, and this is part of the pathway of building the DNA. This enzyme is involved in that, and the para-aminobenzoic acid is an intermediate along the pathway to make those building blocks. So early on I told you, whenever we want to look for something that can be used therapeutically, we have to figure out something that's different about the target organism or the target cells, if you're thinking cancer, or other types of cells than our normal cells, because we need some therapy, some therapeutic agent that can be used that will interfere with the process in those cells that we're trying to kill and not normal cells. And so we're always looking for, it's called relative toxicity, and there's other words that describe it too, but something that's toxic to the bacterial cells, if that's what we're talking about in this case, or the cancer cells if we're talking about cancer, and not toxic or as toxic to our normal cells. And so this is an example. Our cells can't do this process. We don't have this enzyme. So this is a beautiful target. That's why sulfa drugs are great if you're not allergic to them, because many bacteria have the ability to do this, and we can target that process. Our cells don't have the ability to do this. We don't have this enzyme, so we can't do this process. So on this inside of the cell, this is a normal process. Para-aminobenzoic acid is being grabbed by this enzyme, and it's being converted to the product that results after that enzyme catalyzes the reaction. So that's beautiful. The bacteria are doing it every time you put them in the E. coli in the test tube, and a number of the other organisms, they're doing that process so they can make nucleotides so that they can be able to make DNA and be able to grow and survive. We can't. We have to get the building blocks in our diet. So we eat the building blocks in our diet to be able to get those to be able to do this process. So you've got a bacterial infection, and the doctor says, no problem, we'll just give you this sulfa drug, and it's an antibiotic, and it's going to kill the bacteria, and you'll be better in just a few days. So the doctor gives you the sulfa drug, and the sulfa drug is designed to mimic this para-aminobenzoic acid. It's designed to look a lot like it. But look what happens. Here's the sulfanilamide. It's one of the sulfa drugs. There's a whole family of sulfa drugs, and you can see the similarity down at the bottom. It's got the benzene ring, and it's got that para-amino group at the bottom, and that's the part that interacts with the active site of the enzyme. But then look at the top half. It's got this sulfur group with a couple oxygens on it, and then it's got another nitrogen at the top. And so that part looks very different from this carboxyl group that's on the other para-aminobenzoic acid. So you can see we've started to mix the sulfanilamide in. That's these red guys over here. You started taking the drug, and the doctor's giving it to you. You take it 3 times a day, and pretty soon it goes in and it binds the active site that is of the enzyme that's going to catalyze this reaction. And it binds the active site because it looks a lot like the para-aminobenzoic acid. And it fits nicely into the active site. And it gets down in the active site, and it causes that induced fit, that conformational change. And now it gets down in the active site, and the enzyme says, get out of my active site. You're not supposed to be there. I'm supposed to catalyze a reaction, and you're supposed to leave my active site. But the sulfanilamide is different in structure at the top end, the way they depicted it, than the para-aminobenzoic acid, and the enzyme can't catalyze that reaction. And the same affinity that brought that sulfa drug, the sulfanilamide, into the active site is still holding it in the active site. And so the bacteria can't make the precursors that are necessary to make DNA, and therefore it can't grow. And it sits there and says, I want to grow, can't do what I need to do, so I'm going to sit here. And after 2 or 3 days on that sulfa drug, you start feeling better. And you say, I'm good. Don't need to take this antibiotic anymore. 7 days left, 5 days left. Usually they give a 10-day dose, and here in the U.S., overseas they usually give 7 days because that's long enough. But they usually give a 10-day dose here, and so you took 3 days of the dose, and you're feeling great. You're out studying with your friends again, spending all your time looking at books, and you just forget about those antibiotics. And what happens to the concentration of that sulfanilamide in your... That's what you do in your spare time. I know you do. What happens to the concentration of sulfanilamide in your body when you stop taking it? It decreases. And if it decreases, pretty soon that sulfanilamide that's down in that active site, because now there's very little of it and maybe even this much, pretty soon the sulfanilamide that was in the active site is going to dissociate. Everything has a dissociation constant. The interactions between the sulfanilamide and the enzyme are pretty strong, but it's going to dissociate at some point, and when it dissociates, there's not a whole lot of it in the environment anymore to go ahead and fill up the active site, and now the enzyme can run again and do the reactions that it could do. And so the bacteria sat there and said, oh, thank you, you're an awesome student, you forgot to take the rest of your antibiotics, and I'm still alive, and now you're going to have a relapse with the same organism again because you didn't finish your antibiotic treatment. So that's competitive inhibition. That particular one, sulfanilamide, isn't bactericidal, it's bacteriostatic, but pretty soon the bacteria is going to sit there and it's not going to be able to do other metabolic processes because it can't do this one. And so if you take the drug long enough, the organism will die, but over a course of 5 days, 6, the organism, many of them aren't dying. They're just not able to synthesize those precursors to go on and do what they need to do. The other reason that bactericidal antibiotics work well is it slows down the division of the organism, and it gives the immune system enough time to come and gobble those guys up and clear them from the system. Slows down their replication so the immune system can catch up and eat them and clear them from the system. Anyway, that's how a competitive inhibition works, and you can think of a lot of ways to do competitive inhibition. If you can figure out something that looks close enough to the normal substrate that it can trick the enzyme and allow it to bind the active site and block that active site so it can't catalyze that reaction in future steps. So there's also a process called non-competitive inhibition. And this one is kind of like those allosteric, but allosteric is a natural process. It's something that happens in the cell. I'm sorry, I can't get out of your way this morning. So this is something where you put chemicals into the system that actually interfere with the process. And sometimes we use metals that do that. Sometimes we use other types of chemicals that, again, change the conformation of the enzyme. A lot of these are called enzyme poisons, and many of them bind specifically to certain enzymes and block the function of those enzymes. Some of them, like some of the metals, actually oxidize sulfhydryl groups on that amino acid cysteine. Do you remember it? It's one of those 20 amino acids, and it's one that I said you probably want to remember because it's got a sulfur group and it can cause those disulfide bonds that lock an enzyme in position. We talked about them when we talked about the hyperthermophiles and the thermophiles, how our enzymes would denature, but those disulfide bonds hold them. Well, these poisons oftentimes interact with those sulfhydryl groups or disulfide bonds, and they break those bonds, and then the protein denatures, and so it kills the protein. And there's some more description down here at the bottom of the page about those guys as well. But that's a non-competitive inhibitor. Doesn't compete for the active site. It functions kind of like an allosteric inhibitor, but many of these have multiple sites on the protein. So you guys might remember I was talking about crowdsourcing. It's been 3-4 weeks ago, maybe 5, and we're talking about the AIDS, one of the AIDS enzymes that's needed for the life cycle of AIDS, and they were trying to figure out the three-dimensional structure of that enzyme that's involved in the life cycle of AIDS, and all the supercomputers couldn't do it, so they threw it out at humans that they had trained to fold proteins, and that's what they wanted to do. They wanted to find a poison specific for that enzyme that could block the life cycle of the AIDS virus. And once they threw it out there, you already know the story, they figured out what the structure was, and then they could start to look specifically at chemicals that would interfere with that particular enzyme, and hopefully that enzyme alone, so that they could then have some chemical they could throw at AIDS patients and block the replication of the AIDS virus and the devastation that it does. Okay, so we're back at the central metabolic pathways. I think I told you that part of the chapter is designed to let your brain cool down. Everything that you're going to hear now for the rest of the chapter, pretty much you already heard, but in a much watered down form. So now we're going to get some nuts and bolts as we're going through. And this comes to some of those Learn Smart questions that you guys were asking about earlier. So in the central metabolic pathways, we get some major products. ATP is one of those. We get some reducing power in the form of these three guys. Remember, this NADPH comes out of the pentose phosphate pathway. NADH comes out of glycolysis, the transition step, and TCA, and FADH2 comes out of the transition step. So we also get precursor metabolites. Those are those building blocks to make those macromolecules that we were just talking about along the way. And in the central metabolic pathway, it starts with glucose, and glucose molecules can go down different fates, but ultimately they're going to be oxidized to CO2, and then ATP is going to be produced as that process. But if a cell's growing, a cell needs much more than ATP, and many times it's going to pull off some of the carbons from glucose as it's going through the pathway, and it's going to make these molecules that are called precursor metabolites. Remember, I told you table 6.2 and figure 628. You really, really need to know for the final exam. Don't make me cry when you turn that in and you lose 5 or 10 points on the final exam score. I'm telling you multiple times, you're very, very likely going to see it there. So make sure you know about those precursor metabolites from that, those figures. We're going to look quickly at glycolysis. I think you all have memorized it already. You can come tell me afterward if you haven't, and I'll help you start memorizing it. But you know glycolysis starts with glucose, and it takes glucose, and it breaks glucose down to pyruvate. You already told me that. It takes a 6-carbon molecule, breaks it down to 2 3-carbon molecules that we call pyruvate or pyruvic acid. Along the way, there's a couple things. To get started, we have to use some ATP there and those two steps. So like everything, there's an idiom in the English language that takes money to make money, and it takes energy to make energy. It takes ATP to make ATP. And we're using ATP here, and we're using ATP here. Then if we go down to the next step, we've got fructose 1,6-bisphosphate right here at this step. That's a 6-carbon molecule, and there's 2 phosphates, one on the 1 carbon and the other on the number 6 carbon, and that's broken into 2 3-carbon molecules. One of them is glyceraldehyde 3-phosphate, the other is dihydroxyacetone phosphate. And by the way, all of these rectangles, the intermediates that are in rectangles, those are precursor metabolites. Those are the ones that you want to know. those are the ones that you want to know come out of glycolysis as we're talking about them now. So if they've got rectangles in this figure, those are ones that you want to be intimately familiar with when you get to the final exam. So we've got dihydroxyacetone phosphate, and it's turned into glycerol, which is the backbone for lipids. And so this guy can go out to lipid and biosynthesis. And if it doesn't get pulled out for lipid biosynthesis, then it can be turned back into glyceraldehyde 3-phosphate just through a rearrangement. And the next step's an interesting one. The next step needs NAD plus, this step right here, needs NAD plus as a cofactor. NAD plus is the oxidized form of NAD. And it's needed by this enzyme as a cofactor because NAD plus is going to be reduced. That means the chemical that's feeding into it, the glyceraldehyde 3-phosphate, is going to be oxidized in that reaction. So you've got that happening, and you actually get some NAD plus coming out, and you get 2 of those, and each one of those can be used in the electron transport chain to make 3 ATP. And I think that's how they got the answer to the question you were asking about earlier, where they said 8 came out, because there'd be 3 coming out over here if we're talking total ATP, and 3 coming out over here. And then we make a couple right down here in the end. So I'm pretty sure, without seeing the exact question, that's what they were asking about if the answer was 8. Okay? And so we, at the beginning, used 2 ATP. We just made some NADH here. And look at this step. This step actually makes ATP. We get 2 ATP out. So finally, we're even. Finally, we're even on ATP production. We're at a net gain of 0 because we used 2 here, and we just produced 2 here. It's not till the very last step of glycolysis that we get those 2 ATP out that glycolysis makes. So I'm going to switch up just a little bit here and try to go to this guy. And you know what? I just spaced it. Forgot what I was going to do. Sorry, my brain's kind of scattered. Oh, I just remembered what I was going to do. This is all recorded for memory purposes. So, what's the process of ATP production? There's a name for the process of making ATP in glycolysis.
Phosphorylation?
Yeah, all of those that make ATP have that word phosphorylation at the end. And you learned about 3 types of phosphorylation that produce ATP early on in the chapter. So in glycolysis, it's substrate level phosphorylation, right? And so normally I do this on the board, and that's why I got lost for a second. So anytime you have some organic molecule that has a squiggly line and a phosphate group on it, that phosphate is attached by a high energy phosphate bond. Okay? And so if you can take that and you can add it to ADP and move that phosphate group off of there and put it on here, and then you can make ATP, that's substrate level phosphorylation, right? Why do we call it that? Why do we call it substrate level phosphorylation?
Because you're taking one of the substrates and adding it to the ATP to recharge it.
Beautiful. This chemical right here is functioning as a substrate in this reaction. Remember the definition of a substrate? A chemical that feeds into a reaction. And that chemical that I just circled in black is functioning as the substrate of that reaction, and it's carrying this high energy phosphate that can be transferred to ADP to make this ATP. So anytime you see that happening, an organic molecule carrying a phosphate over that's a high energy phosphate bond and is being used to charge ADP back to ATP, it's substrate level phosphorylation. Come on. Okay, so that's what we have happening right at the bottom of this figure. Right here is substrate level phosphorylation. This is also substrate level phosphorylation, and I know you can't see the squiggly lines there, but if you go look closely on the book, the phosphate group here is going to be transferred to ADP and make ATP. and the phosphate group that's right here is going to be transferred to make ATP out of that. So go look at those guys, and that's how the process is happening in the middle of glycolysis. The pentose phosphate pathway is an interesting pathway. I think I already told you that there are some human diseases that if you don't have enough NADPH, then you likely will abort as a fetus. If you make it through birth, then you're going to be very stunted and you'll need to consume NADPH for the rest of your life, and that's because of a mutation in this pathway, and those individuals can't make enough NADPH to function as a coenzyme for those enzymes that need it to power the reactions that they do. Other intermediate or precursor metabolites that are produced out of the pentose phosphate pathway are this ribose 5-phosphate. You've already told me that's used for DNA and RNA. And then erythrose 4-phosphate can go out to be used for amino acid biosynthesis. And you already know NADPH is a reducing power for many enzymes and biosynthetic processes. Did I have you turn and look at Appendix 6 in the back of the book last time? Yeah. So you know that the pentose phosphate pathway is a very complicated pathway, and I spare you trying to memorize that in this class. So, but just know that what's going on there. Glucose 6-phosphate feeds out, and then glyceraldehyde 3-phosphate comes back into glycolysis, and you can look back at glycolysis and see where those processes happen. So the next part is the transition step, and technically this is the transition step right here with pyruvate going, having a carbon kicked out, and then the 2 carbons that remain, the acetyl group, being attached to coenzyme A. And in the transition step, you get some NADH produced, and again, that can go over to the electron transport chain and can be used to produce ATP if the organism has an electron transport chain. All right, so, and then the transition step feeds into the TCA cycle or the Krebs cycle or the citric acid cycle, and as that happens, the 2 carbons are transferred from acetyl-CoA, and then those 2 carbons are fused to oxaloacetate, and now we get a 6-carbon molecule called citrate. And citrate is the reason sometimes that this pathway is called the citric acid cycle, because another name for citrate is citric acid. Okay, citric acid is 6 carbons. It goes through a rearrangement, and then the next step you get a CO2 being kicked off, and now you're down to 5 carbons. That's alpha-ketoglutarate. That's one you want to know because it's in a box. Alpha-ketoglutarate is a precursor metabolite. It can be used to produce amino acids. You get some more NADH coming off at the next step, and look at this. Coenzyme A comes back in right here, gets attached to alpha-ketoglutarate again, and now you've got succinyl-CoA, a succinyl group attached to coenzyme A, much like you saw the acetyl group attached to it on the top of this. And then down at the bottom of this, you see something that the book is going to tell you through a series of complex steps. You get substrate level phosphorylation here. But what happens is that the succinyl-CoA is used, it's phosphorylated, that's a high energy phosphate bond, and then that high energy phosphate bond is transferred to some cells ADP to make ATP. But other cells don't do it exactly that way. Some cells use that high energy phosphate bond and do a different type of substrate level phosphorylation. They actually phosphorylate GDP first and make GTP. So the base on the molecule is different. That's the only thing that's different between them. They make GTP, and then the GTP has the energy to be able to be used by a second enzyme to make ATP. So that's why they don't tell you about the exact process here that's going on. Coenzyme A comes back out. The next step gives us more reducing power and FADH2. And we have then water being put back in, and we get some NADH being produced here. And now we're back to oxaloacetate again, a 4-carbon molecule. And I hate to stand behind here, but I also keep realizing I'm blocking you. So 4-carbon molecule that's going to be fused to the 2 carbons that are being carried back in on acetyl-CoA. And then the cycle goes around again. Keep in mind, for every molecule of glucose that feeds in, there are 2 pyruvates that are produced. So this cycle happens not once, but twice if all of the carbons come through the system. But if any of the carbons get pulled out, for example, if this pyruvate's used to make amino acids, which it can be, then none of the carbons make it any further than that. The pathway's blocked right there. So if pyruvate's used to make amino acids from that particular glucose, the only ATP that you got was up in glycolysis. Does that make sense? Same thing for acetyl-CoA. If you pull acetyl-CoA out, it can be used to make amino acids as well. Then the rest of the carbons don't go through this pathway, and it's blocked as well. Or if oxaloacetate is pulled out to make amino acids, which it can be, then it's going to be blocked, and the pathway won't go any further than that. And by then, a lot of energy has already been pulled out in the form of all that reducing power that came out, as well as the ATP that was produced. Likewise, if this acetyl group gets pulled out, where are acetyl groups used? What kind of molecules are they used to build? Lipids, beautiful. Which part of lipids? This is all in chapter 6 in the test, so I'm trying to prep you for the test. Which part of lipids are going to be made out of that acetyl group? Not the backbone. What is the backbone, though, since we got there? The backbone. Glycerol. Remember, it comes from that dihydroxyacetone phosphate that I pointed out to you earlier, right after the branch. So these guys are used to make these. Hopefully there's enough room there that you can see it. Whoops, I just cleared it. What do you think that stands for? Fatty acids. Beautiful. So fatty acids are usually multiples of 2 in length. You might get an 8 carbon fatty acid, a 10 carbon fatty acid, 12, 14, 16, 18, and so on. Almost all of them are even number lengths, and it's because those 2 carbons are being pulled off of acetyl-CoA and sent down the biosynthetic pathway to make fatty acids. Beautiful. So we get reducing power, and you already know from anatomy and physiology that that reducing power then can be pulled over into the electron transport chain, and you can get lots of ATP. That's what those questions in the Learn Smart are asking you about. The guy that figured that all out got a Nobel Prize back in '78. It's now called the chemoosmotic theory, meaning that everything is balanced and those chemicals can be produced to feed into other pathways to drive those pathways. The electron transport chain that you learned about in anatomy and physiology is very, very similar to the system that many bacteria use. There's just a few differences, and I'll point those out in just a second. But they all basically are embedded in membranes, and basically you need the membrane to make a barrier. The membrane is a barrier. Remember, it's semi-permeable. And what you do is pump protons out, and when you pump protons out, then you've got a concentration of protons on the outside that's high and a low concentration on the inside. And the driving force for those proton pumps is electrons. And so NADH, FADH2 carry high-energy electrons over to those proton pumps, just like your battery does. Once it's charged up in the morning, it delivers high-energy electrons to all the circuits in your phone, and these electrons drive the circuits that are part of the electron transport chain. The electrons pass through each one of the circuits along the way, and a little bit of the energy is pulled out of those electrons as they're going through those circuits. And so they go from a high-energy electron and hit one circuit, a little bit of energy is pulled out. They might hit another circuit and another one, and gradually you get down to what's a low-energy electron. Okay? Still a negative charged electron, but the energy level of that electron is a lower energy electron. It's got to be dealt with because it's there and it's at the end of the electron transport chain. It's got to go somewhere. And the somewhere that you know about from anatomy and physiology is for an oxygen molecule to take that electron, and when it takes it, then the oxygen becomes negatively charged. It takes 2 electrons, so it becomes a 2 negative charge, and nothing wants to be charged typically in nature. So when oxygen is sitting around as an oxygen atom and it's got 2 negative charge, it's in solution, and so there's protons around. It's going to pick up those protons and it's going to become water. Okay, then it's neutral, relatively neutral. So that's the process of how the electron transport chain works. That should be pretty familiar to you. And the system in eukaryotes is very similar to the system in prokaryotes. It involves all of these electron carriers and molecules that deal with moving electrons and pumping protons outside of the cell. Ultimately, we get proton ejection outside of the cell. Hydrogen passes through, and the electrons are being used as a power to drive that. You have to have that concentration gradient to drive this process of actually finally making ATP in the end, and that's provided by electron transfer that comes through the system, through those carriers that we talked about. In the mitochondria, you hopefully remember this from anatomy and physiology. You've got a complex 1, 2, 3, 4, and then some terminal oxireductases at the end of that process. And you all remember that from anatomy and physiology. I'm 100% sure, except for one of you hasn't taken it yet. So I'm not going to spend a lot of time talking about that, but what I do want to do is look at the process that's in the bacteria, because it's pretty analogous. And first we're going to look at E. coli, because E. coli does things a lot like your mitochondria do. your mitochondria, we didn't understand the process of what they were doing until people started studying E. coli and figured out these processes in E. coli. Then they looked at the mitochondria in our cells and said, hey, they do it a lot the same. And so E. coli is capable of aerobic respiration. That's what your mitochondria do. That means that oxygen is used in the last step. Remember that last step? Oxygen takes the terminal electron, takes it becomes charged -2, and then you get water being produced, and that's what happens when you have aerobic respiration. So E. coli has two different types of NADH dehydrogenases. They're just going to show you one, but they're pretty analogous to complex 1 in the mitochondria. They function as a proton pump. They pump protons outside of that membrane. And then E. coli has a succinate dehydrogenase that's equivalent to complex 2 of the mitochondria. And then there's some alternatives that can happen and several different ways that E. coli can do it. But E. coli lacks the complex 3, and it also lacks cytochrome C oxidase. Somebody was remembering, I forgot what it was, something Mo. What was your acronym for?
Child of Mo.
Child of Mo. And so one of those is cytochrome C oxidase, the oxidase test. Must be the O in Mo. Yeah, and that's what you're actually looking for. You're looking to see if the organism has cytochrome C oxidase or not. And that's the oxidase reagent that you're looking at. And so that's part of this process that we're talking about, the electron transport chain. Okay. Ultimately, the electrons are coming through the system, and we just talked about it, and they're bouncing off of these molecules, and finally at the end you get a low-energy electron that's got to go someplace, and in aerobic respiration that's going to be picked up by oxygen, which will then ultimately be converted to water. So E. coli can also do respiration via anaerobic respiration. And in that case, it means there's no oxygen around. If there's no oxygen around, something else has to take those electrons that just came through the electron transport chain. And E. coli can use a number of things. It can use nitrate, and it's converted to nitrite. It can be a lot of different things that can be picked up and use the terminal electrons that come through that process. And then others use even some sulfur-containing compounds as terminal electron acceptors. And in that case, they produce hydrogen sulfide as an end product. So there's lots of different ways that bacteria can do the electron transport chain. Here's a picture of how it works in E. coli in the general sense. And we've got this NADH dehydrogenase. Remember, E. coli has a couple of different molecules that can do that. The name tells you one thing it does. It dehydrogenates NADH. It kicks hydrogens off of NADH. And you already knew that because the mitochondria does the same thing. Those hydrogens in this case are fed by NADH, but the hydrogens are important, but there's something else that comes out of NADH. What's that? Electrons, right? NADH is an electron carrier. It's bringing electrons or electricity, right? Driving these lights, driving this enzyme. So the electrons that come out of NADH are providing the power to drive this enzyme. So ultimately, its real goal is to pump these protons out of the cell so you can get that proton gradient and have a proton motive force. So that happens. NADH dumps the protons. The electrons follow and are used by this enzyme to charge it. Those protons are pulled out and dumped outside of the cell. Okay? So we just got a gradient charged a little bit there. The electron comes over, it hits this chemical called ubiquinone, and then ubiquinone just passes the electron over to the next proton pump that's going to pump some more protons outside of the cell. And then the electron is relatively low energy. It's picked up right here at the end by oxygen, and we get that water being produced again. Okay, so for every NADH that feeds into the system, you get approximately 3. In one place the book says 2.5 plus, and it's all stoichiometry, and it's a little bit complicated. So 2.5 to 3 ATP that comes out of the process when an NADH feeds in. But FADH2, you probably learned this in anatomy and physiology too, when FADH2 feeds into the system, you only get 2 ATP being produced. Did you figure out why that was? FADH2 cannot feed its electrons into this enzyme because that's NADH dehydrogenase. And that's part of the reason I want you to go look at those 3 molecules to see what's the difference between them. FADH2 feeds its I'm just going to abbreviate F here. It feeds its electrons into this succinate dehydrogenase. It's in the textbook. It's way down in the fine print under that figure. I don't think it mentions it anyplace else. So if you're not reading the figure legends as you're going through the chapters, you're missing lots of good nuts and bolts to help you understand. The electrons from FADH2 feed in right here at succinate dehydrogenase, and it misses this first proton pump. The electrons never hit that pump. They start right here. They go from succinate dehydrogenase over to ubiquinone, and they're transferred to ubiquinone, and then from ubiquinone they're transferred over here. They drive this proton pump, but they miss the first proton pump, and so you only get those protons being pumped out, and that's why you get less ATP ultimately produced from FADH2 compared to NADH. Okay, so this portion of this figure right here is the electron transport chain. And that's what you learned about in anatomy and physiology. And then you went on to say this proton gradient is being built up here with lots of protons up here in this area. And down inside of the cell, there's not very much protons, not very many protons down in the cell. Okay, and that's a gradient. That's a battery. And you learned about in anatomy and physiology that gradient being able to drive this enzyme that looks very much like it does in E. coli called ATP synthase. Those protons can be allowed to feed back through this enzyme, and as they feed back through this enzyme, they're carrying electrical charge. They're charging that enzyme so it can do something else. And that something else is to be able to take ADP and add a phosphate group onto it and make ATP. And you know that this process is called blank phosphorylation. What's the blank? Oxidative phosphorylation. So I ask this question every semester. I think you guys are much smarter than most students, and if you're listening from a previous semester, it's not really an insult, but these guys are smart. I think you guys are much smarter than most students this semester, and all my classes are. If you're listening to this from one of the other classes, don't be offended. Why do we call this oxidative phosphorylation? Prove me right.
You're taking oxygen away when you're phosphorylating?
You're taking oxygen away right here. Is that what you're telling me?
Yeah.
Yeah. If you take oxygen away, is that oxidation? No. Water's actually, what's happening to water, or the oxygen in that case? It's being reduced, right? Electrons are being added to it. It's being reduced. So you never asked this question when you were in AMP? You never thought about it? Why do we call it oxidative phosphorylation? No wonder it didn't make sense. No wonder you don't remember this process. Somebody's got to know, right? I just said you guys are smarter than the average student. Prove me right. Why do we call this oxidative phosphorylation?
Okay, what was it?
It's right over here. What starts the whole process? What kind of reaction is that? What kind of reaction is this right here? That's an oxidation reaction. And when FADH2 feeds its electrons into succinate, that's also an oxidation reaction. And that's why we call this oxidative phosphorylation. Directly, it has nothing to do with this enzyme. But the whole process has to be linked together, or else that enzyme can't do its process. So this is oxidative phosphorylation, and the reason for it is right there at the beginning of this process. NADH is being oxidized to NAD+, and FADH2 is being oxidized to FAD. When we come back on Friday, we're going to finish this chapter, and we're going to talk about active transport. I don't really have to talk about this one because we already talked about it way back in chapter 3 or 4, and if you don't remember it, you should go back and look at chapter 3 and 4, but I'll talk about it briefly. And we talked about the second one, the movement of the flagellum, way back in chapter 4, so I really don't have to spend a lot more time on this figure when we come back on Friday.